Hello everyone. This week in lab, we're going to go ahead and talk about control of microbial growth. We're going to discuss the chemical methods. Now, for details actually about the topics that we're about to discuss, I would refer you back to our lecture video. So watch the antimicrobial video. There were two antimicrobial videos that we talked about in the lecture, part 1 and 2. So those will definitely have way more details compared to, this video right here. So when we think of, physical control methods of limiting the growth of bacteria, we can think of, for example, putting, our agar plates in the autoclave machine. So we can think of the autoclave machine as one, right, as a physical control to go ahead and kill the bacteria. And the autoclave will expose the bacteria to high heat and at the same time lots of moisture, right? Lots of humidity, and that's how we can go ahead and kill the bacteria. But I mean, there are limitations of using the autoclave machine. If you guys think about it, if you get infected with a bacteria, would we go ahead and place you in an autoclave machine to kill that bacteria that has just infected you? Absolutely not, right? So we go ahead and use like other methods, such as like antibiotics, etc. So again, there are, there are situations like this, okay, where the autoclave definitely will not be used. And then even with equipment and, certain supplies, we cannot stick them in the autoclave because again, they will actually be destroyed. So again, things that have like powder and oils would not, we cannot stick them in an autoclave machine. They will get destroyed. And when we also think of, UV light, and you would think like, okay, I can use UV light to go ahead and kill the bacteria. Again, there are some limitations of using the UV light. And that is one thing is you need direct exposure because UV light doesn't really pass that well, through certain, material like, for example, plastic. And so you would have to have direct exposure to be able to kill the microbe. And then, of course, as if we think of us humans, we don't really want to be near also like, high concentrations of UV light because it could definitely, cause us harm. Okay. There are some terms that you need to be familiar with, and again, we've discussed those terms in the lecture. Disinfectants, we talked about how disinfectants are basically products that are used to decontaminate, inanimate objects. So you cannot use a disinfectant to disinfect yourself because these chemicals are usually pretty harsh, okay, to be used on your, you know, on your skin, for example. So again, you would have to use them on inanimate objects like, surfaces, tables, etc. Antiseptic, those are products that can be used on the skin and because they're not going to be as harsh, okay, on your skin. And we said that antiseptic, if you remember, we said that antiseptic are not going to sterilize our skin, right? So it's just going to reduce the number of bacteria that's going to be on our skin, but it's definitely not going to sterilize our skin because it's still not going to kill all the microbes and it's definitely not going to kill the endospores. Broad spectrum, those are the drugs that can actually target like a variety of microorganisms, those that are gram positive and gram negative. Narrow spectrum, those are drugs that will only target one specific group, so either gram positive or gram negative. Bactericidal, whenever you see the suffix cidal, those are the drugs that are going to kill the bacteria. Bacteriostatic, whenever you see the suffix static, those are the ones that are going to inhibit the growth of the bacteria, but they're not going to be killed. It's just going to slow them down. Okay. Now, if we talk about, some chemical control of the microbes, we can talk about the disc diffusion test. And if you recall, we also, refer to it as the Kirby-Bauer method. So you can refer to it as the Kirby-Bauer or the disc diffusion test. And that's when you go ahead and examine the zone of inhibition that's going to be around that disc that has the disinfectant, okay? So, by the way, you can do this test with discs that have different disinfectants. So this could be alcohol, this could be bleach, chlorine, etc. Or those discs could be antibiotic discs. And so you would be testing penicillin versus tetracycline versus, erythromycin, etc. And what you want to see is the zone of inhibition around these discs. If there is no zone or if the zone is very, very small, I mean, this would give you an indication that the bacteria is, it's resistant, okay? It's resistant to that disinfectant being used. Versus if you see right here, the large zone of inhibition, you can tell that this disinfectant is doing a better job. But for you to know whether the bacteria is susceptible, intermediate, or resistant, as we discussed in lecture, you can't just go with whether there is a large zone or a small zone and that's it. You have to measure the zone and then take a look at the chart and figure out whether your bacteria is susceptible, intermediate, or resistant. So as you can see right here though, like if there is no zone of inhibition or if it's very, very small like this, we can tell right away that this chemical, whatever chemical you've used right here is not effective. The bacteria is resisting it. Definitely, if you see a larger zone, then the chemical is more effective. But again, you still then, I mean, if you want to compare, let's say, those two, you have to go ahead and use the chart to figure out which disinfectant is better. And one more thing to note is when you go ahead and place that disc, which is impregnated, let's say with an antibiotic, or it's impregnated with a certain chemical, let's say, for example, bleach, okay? As the chemical diffuses out onto the agar, the concentration of that chemical reduces as it diffuses out. So as this chemical right here diffuses out onto the media, you can see right here, kind of like, with the colors, the color is fading, and that would mean that the concentration of that chemical is actually reducing as it moves away. And so there is something called the minimum inhibitory concentration, or the MIC. And that is basically what is the minimum amount of the chemical that's going to be necessary to kill the microbe. And so it would be like out here, the minimum inhibitory concentration that can go ahead and kill, the microbe. And we said, though, I mean, from the disc diffusion method or the Kirby-Bauer method, you can tell that the MIC is going to be like out here, right? This is where you have the minimum effective, basically, inhibitory concentration. But for you to get an actual number for that MIC, you will have to do the other tests that we've discussed in the lecture. Either do, a tube dilution test, or do the E-test or the MIC assay for you to get a specific number. But from this right here, the disc diffusion test, I mean, you can tell that, the MIC is going to be further away from the disc, depending, of course, on the size of your zone. The MIC could be pretty far away from your disc, or it could be close, depending on how large your zone is. So if we take a look right here and we take a look at what is the relationship between the minimum inhibitory concentration and the size, well, notice how the size of the zone of inhibition is super small. That tells you that the MIC, the minimum inhibitory concentration for this chemical or for this antibiotic is really high. Because notice how it didn't even diffuse that much. So that means that the concentration of the drug that diffused is really high. Versus the larger the zone, right, you know that the drug had the ability to diffuse further and further away. And remember how we said the more it diffuses away from the disc, the lower the concentration gets. And so we know that the larger the zone, the lower the MIC is going to be. And so therefore we say that the MIC, the minimum inhibitory concentration and the size of the zone, they are inversely related. As the size of the zone increases, right? So right here, as the size of the zone increases, the MIC is basically going to decrease. So that's an inverse type of relationship. So right here, this one is going to have such a high minimum inhibitory concentration. And then this one, because it has a larger zone, it has a lower minimum inhibitory concentration. Now, do know this table, okay, guys? You do need to know the category of these chemicals. So some of the chemicals that we use are known as halogens, and these are some examples like chlorine, which is basically bleach, and iodine, and that is a broad spectrum. So it can target both gram-negative and gram-positive. So that's why, we use bleach a lot in our microbiology classes because it is a pretty good, broad spectrum chemical. Heavy metals, those are, chemicals that have things like gold or silver or mercury, these metals. And again, those are broad spectrum chemicals, and so they can target, again, gram-positive and gram-negative. Oxidizing agents, for example, hydrogen peroxide or peracetic acid, again, they are also broad. Phenols and alcohols, things like 70% isopropyl, what we use, ethanol, those are narrow spectrum and they target gram-negative. And what they do is they basically dissolve the OM. What is the OM? The outer membrane. If you recall, we said that the gram-negative bacteria, they have their plasma membrane, and then they have their cell wall, and they have that outer membrane. So the phenols and alcohols, they go ahead and they break down that outer membrane, and they're and that's how they kill the gram negatives. The quaternary ammonium, or we refer to them as the quats. Most of the household cleaners, by the way, are under this category right here. And they end in ammonium chloride, and those are narrow against the gram-positives. So those are narrow spectrum, and they target the gram-positive bacteria, and they basically have the ability to go through the cell wall of the bacteria. So they are narrow spectrum targeting gram-positive bacteria. So you guys, as I said, a lot of times you're going to see the test names for the Kirby Bauer and the disc diffusion used interchangeably. Some refer to the disc diffusion test when you use the discs that are impregnated in chemicals like chlorine and bleach, etc. And then some use the Kirby Bauer, they refer to it as Kirby Bauer when the discs are impregnated with antibiotics. But again, some just use it interchangeably, whether that disc is impregnated with bleach or antibiotic. You would find some textbooks, etc., referring to the whole, to any of that, test when the antibiotic or the chemical is being diffused out. They can refer to it as the Kirby-Bauer test. Now, with the Kirby-Bauer, and let's say those discs are definitely impregnated with antibiotics, we have to use this type of plate whenever you are using antibiotics, okay? Now, if you're not using antibiotics, and let's say these these discs are impregnated with different chemicals like iodine, bleach, chlorine, etc., then you can just use, by the way, a regular nutrient agar plate. But if you're going to use antibiotic impregnated discs, you must use this type of plate right here, the Mueller-Hinton agar plate, because the antibiotics are going to have an easier time actually diffusing within this medium, okay? And that's why we use it. And what you have to do is go ahead and you grab that Mueller-Hinton agar plate. You go ahead and you inoculate your plate with the bacteria. And you have to usually inoculate the plate like several times, like 2 to 3 times. So you would grab your inoculating loop, dip it into the sample, and then you would go ahead and, zigzag method, once, and then you would go ahead, turn the plate like 60 degrees, 90 degrees, and then you would go ahead and in the opposite direction, you would go ahead and inoculate, so second time, and then go ahead and rotate a third time, and again, go in a different direction and go ahead and inoculate a third time. And this just ensures that you didn't leave any spots, okay? You've covered the whole entire plate, and this is what we are, eventually we're going to look for, that confluent lawn growth. After you go ahead and you inoculate your bacteria, by the way, just gonna wait a few minutes and then go ahead and dispense the antibiotics onto the plate. And then after you go ahead and dispense the antibiotics on the plate, you can go ahead and incubate this plate in the incubator. Now, after you take it out of the incubator, you go ahead and you start measuring the zone of inhibition using a ruler. So you would go ahead and measure the whole entire diameter. Okay, so measure the whole entire diameter. And as I said, you have to, first of all, measure it in terms of millimeters. Okay, so measure it in terms of millimeters. And you're going to take that number, and you're going to be given a chart, and you have to compare your number to the chart that you are given to figure out whether your bacteria is sensitive to that antibiotic, or whether it's intermediate to that antibiotic, or whether it's resistant to that antibiotic. Resistant to the antibiotic, meaning your antibiotic is not working at all against it. Intermediate, meaning your antibiotic is working, but, it's like medium effective. And then sensitive, that means your antibiotic is working very well against it. Let's take a look at this table right here. It lists a bunch of antibiotics for you. As you can see, antibiotics can target several different things. So let's talk about this group right here. This group of antibiotics, they target the cell wall of the bacteria. So basically, they prevent the production of the cell wall. So they inhibit the production of the peptidoglycan. And these antibiotics, they are narrow spectrum, so they work against the gram-positive bacteria. They're cidal, so they're going to kill the bacteria. These antibiotics listed over here, those are going to target the protein synthesis. They're going to inhibit the protein synthesis. How do they do that? Well, they go ahead and they attach themselves to the 70S ribosome within the bacteria. And by attaching themselves to that ribosome, they basically block the production of proteins. Those are also broad spectrum antibiotics, and those are static. So those are going to inhibit the growth of the bacteria. The nucleic acid inhibitors, this antibiotic right here is going to basically inhibit the RNA polymerase from working, and so therefore it's going to block the DNA transcription from taking place. It is a broad spectrum antibiotic, and again, it's cidal, so it's going to kill the bacteria. Some of them, they go ahead and they inhibit the enzyme activities. So there's this enzyme right here, and again, it inhibits, enzymes. As long as you know in general that it inhibits the enzyme, you're good. I'm not going to ask you to know the specific enzyme. And it is a broad spectrum antibiotic. It's static, so it goes ahead and inhibits the growth of the antibiotic, of the bacteria, of course. And then some antibiotics, they target the cell membrane, so such as polymyxin B. So polymyxin, again, it can disrupt the phospholipid bilayer of the plasma membrane. It's narrow spectrum, works against the gram-negative. Polymyxin B, one thing to know about them is they work, by the way, really well against Pseudomonas. Pseudomonas bacteria, for example, Pseudomonas aeruginosa. And those are cidal. They're going to be able to kill the bacteria. So Pseudomonas aeruginosa is a gram-negative, and again, polymyxin B works really well against it. Now, there are three genera of bacteria that are, it's pretty challenging, okay, for us to go ahead and, kill, or, using those disinfections and these, like, chemicals and, the genre Bacillus and Clostridium. Those are two genera that produces endospores. And again, these endospores are pretty chemically resistant, so it's pretty hard for us to kill these endospores. Makes it definitely way more challenging. And then Mycobacterium is another genre of bacteria that are also pretty hard to get rid of because they have that lipid, waxy mycolic acid in their cell wall. Which is, again, that waxy layer that they have, it makes it super hard for the chemicals to be able to pass through it and enter into the cell. A third group is the Pseudomonas genre, and those, they basically have very narrow pores within their outer membrane, so they're gram-negative, and so that's why they have an outer membrane. And so therefore, again, it makes it very hard for the chemicals to pass through, and therefore makes it, again, way more challenging to kill. As I said, though, polymyxin B is pretty effective against the Pseudomonas because it does disrupt the cell membrane. So that's how, again, the chemicals will be able to make it into the cell, and the cell will die, like the bacteria would die. For the virtual lab for this week, you're going to go ahead, you're going to be given different, bacteria samples, and you're going to go ahead and you're going to inoculate the plate. So you're going to go ahead and inoculate first the plate with the bacteria, and then you're going to use the sterile, discs that are basically coated with different disinfectants. And you're going to go ahead using the tweezers, okay? You're going to go ahead and place them onto the plate. You're going to have samples of discs that are, soaked in water, some bleach, mouthwash, iodine, and soap. Okay, and so you're going to go ahead and place the discs equidistant. So equal distances are going to be left apart between the discs, and then of course the plate is going to be incubated so that you can go ahead and start measuring the zone of inhibition. So with the Kirby Bauer test, again, specifically, like, the antibiotic discs that are impregnated, you're going to start with the Mueller-Hinton agar plate. You're going to grab the cotton swab, and you're going to go ahead and inoculate the plate several times, right, with the sample. And then what you're going to do is go ahead, place the discs, the antibiotic discs onto the plate. Now, sometimes labs have those cool dispensers for the antibiotics. So you can see right here several antibiotics and like placed all at once, and all you got to do is like just push on that plunger, and it's basically going to lay down all of the antibiotics for you all at once, which is really great. Um, And right here, you're going to, like, there are a few videos for you to watch just to kind of see how the disc diffusion method and the Kirby Bauer, are taking place. As I said, you guys, a lot of sources out there use those two interchangeably, okay? Um, Now, to be more specific, though, if you use the disc diffusion method, then you're talking about those discs that are impregnated in chemicals. Kirby Bauer, those are going to be the discs that are impregnated with antibiotics. And then, of course, there are the virtual labs, on Connect that you're going to complete. These are some results that I want you to go ahead and take a look at. So let's say you inoculated this plate with Staphylococcus aureus and you've inoculated this plate with E. coli. And you went ahead and you put the discs that are impregnated with the different chemicals for the disc diffusion test. So like water, soap, iodine, etc., bleach. And you're supposed to go ahead and take a look at the zones of inhibition and kind of try to figure out, which chemical is the best effective, least effective, etc. And then this one right here, the Kirby Bauer test, again, you're going to inoculate your plate. So let's say this is a plate that's already inoculated, and those are the discs that are impregnated with antibiotics, okay? So one more time, take a look at the zones of inhibition and answer the questions, whether your bacteria is, let's say, resistant to that antibiotic, susceptible, or intermediate.